Hello friends, today I am going to explain the mechanism of enzyme action. So the enzyme, we all know it is a specific for their action. There is a specific enzyme for every cellular substrate and reaction. If there is a series of the reaction and in series of reaction in every steps there is a separate enzyme is there because enzyme is specific for their substrate. This enzyme is catalyze the reaction and it produces the product. The mechanism of enzyme action, it have the 3 to 4 steps are there. So in first step, the enzyme is acts on the particular specific substrate and the enzyme substrate complex is formed. So this is the first step. Then in second step, the enzyme substrate complex, there is some changes is occur in substrate, not in the enzyme. And in third step, from this enzyme substrate complex, the product is formed. And in last final step, after formation of the product, enzyme is released and again it acts on the another substrate molecule. So here this is a continue till there is a substrate molecules are in the reaction. So this mechanism of enzyme action is can be understood by two hypotheses for understanding the mechanism of enzyme. The Emil Fischer in 1890 proposed a lock and key hypothesis. So this lock and key hypothesis is very simple hypothesis to understand the chemical reaction or enzyme action. So according to this hypothesis, the enzyme have the active site or it is also called as a allosteric site. So this allosteric site or this active site, they are complementary with the active site of the substrate, and this enzyme active site are rigid structure, they cannot change, and when this enzyme active site and this substrate active sites are complementary, then it is the same relation between the lock and the key. That means the enzyme is the lock and the substrate is a key. The key is specific for the lock. Similar way, the substrate is also specific for the enzyme, and therefore this substrate is fit into the enzyme similar to the any key is fit into the particular lock, and then the enzyme substrate complex is formed here, and then the product is formed and the enzyme is released. So here this is called as a lock and key hypothesis. You have to remember the active site of the enzyme is a rigid structure, it is not changed, it is not flexible, and therefore for such active site the specific enzyme is essential for the completion of the reaction or formation of the enzyme substrate complex. So this is called as a lock and key hypothesis. Second one, that is induced fit hypothesis. So induced fit hypothesis is proposed by scientist Koshland in 1966. So this theory explains the enzyme action on the substrate. So according to this theory, the substrate induces, so this substrate is induces to this enzyme, that means some conformational changes are takes place in the enzyme due to this substrate, and the main character of this theory is flexibility of the active site of the enzyme. That means here this active site of the enzyme is not rigid, it is flexible. That means it is changes according to the substrate. That means here this substrate is induces the active site of the enzyme, and this relationship is similar to the hand and glove relationship. So the substrate is acts as a hand and this enzyme acts as a glove. Similarly, here, as like the glue is fit into the small or the large hand, in this, in similar to this, the substrate is changes the structure or active site of the enzyme and it is fit into the enzyme. That means here in this example here, this is the active site, but this active site is not complementary to this substrate, but this substrate it changes this active site and then this substrate is becomes fit into the this enzyme and the ES complex or enzyme substrate complex is formed and this reaction should be completed. So this is called as a induced fit hypothesis. That means the substrate is induces the enzyme. And next point in this unit, that is the factors affecting on enzyme activity. So here for your study, there is a temperature and the pH are the two factor here. So you have the temperature. So in maximum activity of the enzyme is takes place when the temperature is 25 to 40 degrees Celsius. If the temperature is becomes minimized at 0 degrees Celsius, most of the enzymes are inactive, and the enzyme may also lost their activity when the enzyme is increased, temperature is increased, that means when the temperature is 40 to 60 degrees Celsius, then the enzyme is becomes denatured and the enzyme cannot catalyze the reaction. But here, if you take the example of the, if you plot the graph in temperature versus the rate of the reaction, then we will get such bell-shaped structure. So here bell-shaped graph. So this is actually the optimum temperature. So here the optimum temperature is about 25 degrees Celsius to 40 degrees Celsius. So at 0 degrees Celsius there is no slope or there is no reaction, and when the temperature is increased here 5, 10, then slowly there is a rate of the reaction is increased, and when there is more than 30 to 40 degrees Celsius, then here there is a maximum rate of the reaction, that means velocity of the reaction is increased, and when the temperature is increased beyond the 40, that is beyond the 50 degrees Celsius, then automatically the rate of the reaction is becomes less and less. That means here this graph shows that the optimum temperature range for the enzyme activity is 25 degrees Celsius to 40 degrees Celsius. Below that range, the rate of the reaction is becomes minimum, and above that optimum temperature, the rate of reaction is also minimized. That means the temperature is affect on the rate of the reaction. Ultimately, it is affect on the activity of the enzyme. Second one, that is the pH of the enzyme. So here you know the pH of the enzyme. The optimum range of the pH is here particularly in the neutral. That means here neutral pH, the enzyme is activate more active, and enzyme are very sensitive to pH of medium in which they are because their proteinaceous nature. That means here when the pH of the medium is becomes decreased, if the highly acidic, then the rate of the reaction is minimized, and when the pH of the medium is alkaline, more alkaline, that is 10, 9, 10, 11, then also the activity or the activity of the enzyme is decreased. But here in a neutral range, that means 7 or the 8 pH, the activity of the enzyme is increased. That means here 7 to 8, this range is a optimum range of the pH. That means here we say that the pH is also affect on the activity of enzyme. But there are certain enzyme, if you take the example of arginase enzyme. So this arginase enzyme is active even the pH is 10 pH, and therefore such enzyme is very exceptional cases, but in general most of the enzyme they are optimum pH range is 7 to 8. So this is a factors affecting on the enzyme activity. Then here another point, that is inhibition of the enzyme. So the inhibition of the enzyme means there are certain substance or there are certain factor or substrate, they retard, they inhibit the activity of the enzyme, and such substrate or such substances are called as an inhibitor. So this is also the one more property of this some enzyme. So in that there is a two inhibitions are there. First one is a competitive inhibition and another is a non-competitive inhibition. So in competitive inhibition, the competitive inhibition involves a molecule or the substrate which are compete to the another substrate. That means here suppose this is the reaction. So this is the enzyme, and this enzyme is when react with this substrate 1 and this is substrate 2. Actually this substrate is identical to the active site of enzyme, and this another substrate is partially identical to the active site of the enzyme. But here this substrate, here another substrate is compete to this substrate, and then this substrate, second substrate, is fit into the active site of the enzyme, and then it block the reaction. Such substrate or such substance is called as inhibitor, and this inhibitor is compete to the main substrate, and therefore such inhibitor is called as a competitive inhibitor. That means this competitive inhibitor is inhibit the enzyme activity and stop the reaction, and this inhibitor or this mechanism is called as a competitive inhibition. So I think you have got the what is mean by competitive inhibition or inhibitor. So we have again repeat. So suppose this is the enzyme. In the reaction mixture there is this is the substrate, and there is also another substrate is there. So this another substrate acts as an inhibitor, and this inhibitor or this substrate inhibitor is compete to this main substrate, and instead of this substrate the enzyme is acts on this inhibitor, and enzyme inhibitor complex is formed, and when such complex is formed then there is no reaction, the reaction is blocked, the product is not formed, and this mechanism is called as a competitive inhibition, because this inhibitor molecule is compete to this substrate, and therefore it is called as a competitive inhibitor. So in another way there is a non-competitive inhibitors are there, and this mechanism is called as a non-competitive inhibition. So the non-competitive inhibition involves a molecule binding to a site other than the active site. That means here this non-competitive inhibition, the inhibitor is not partially identical. This inhibitor is completely different from this substrate, and this inhibitor have the capacity to induce the active site of the enzyme, and due to their such property this inhibitor is not compete to the substrate. This inhibitor is directly react with this enzyme, and enzyme inhibitor complex is formed, and then there is no specific substrate, and therefore here there is a no reaction is formed. That means the substrate is fit into the active site, but this active site is changed due to this inhibitor molecule, and therefore due to this changed active site of the enzyme is cannot catalyze the reaction. Such mechanism is called as a non-competitive inhibition, and this inhibitor is called as a non-competitive inhibitor. So we can such reaction is we can block by increasing the substrate concentration. So here this is also one more property of the enzyme.